Hello and welcome back to OncoDiaries. So today I am joined by Dominik Brücher, the CTO of CIS BioPharma. How are you, Dominik?
Good to see you, Izzy.
Yeah.
Well, a bit tired, but otherwise very much looking forward to the interview.
Good, amazing. It's great to have you on the podcast. So, as you know, we usually explore cancer through the lenses of science and innovation through lived experience, which Dominik has got quite a lot of that himself. And today we are going to be discussing ADCs versus radiopharmaceuticals. So, to start off with, Dominik, do you mind just giving me a little bit of your background and how you've entered in this space?
Yeah, very much. So my journey was, I would say, more driven by my curiosity about designing proteins. Educated as a protein engineer based in Germany, where I'm originally also coming from, but then also in the US at University of California, Berkeley. And I was always driven by the fascination of building something new, understanding a system, then building new proteins that have an effect on a system. I'm not coming from the biology perspective, but I'm really more coming from the therapeutic modality perspective. And it's a good fortune working on excellent teams in that area at Max Planck Institute and in silico protein engineering, even before the days of OpenAI and the revolution in protein engineering, which is now a very different field. But then also to work on some industry, large pharma corporations like Roche, where we did a lot of innovation in period in Germany on also ADCs, development of new ADCs, linker payload combinations, even back before 2015. So the field is kind of turning around to that. I had also the good fortune of having wonderful educational mentors. I studied or did my PhD here in Zurich in Switzerland under Andreas Plücktün, who is very renowned for funding a lot of companies around protein engineering, body modalities. I was working with gene therapy for there as well, so really driven by the modality then again, and had the opportunity to build up a startup based on venture, a US venture-backed company here in Basel as well, and built it with over 40 people, all driven again in the oncology field, but really on focusing on new protein modalities. And as you mentioned, since a couple of years, I have now the great honor to joining CIS BioPharma as the CTO, leading the research and development teams, bringing our first product to the clinic within the next 2 years.
Amazing. And just listening to you talk about your education there and, the mentors that have sort of shaped you into where you are now, did you always see yourself working at the interface of biology and chemistry or?
Not particularly, I would say. I saw myself very quickly in the protein engineering part, right? So I was never shy about the chemistry, also the chemistry in like organic biochemistry part.
Mm-hmm.
But I never foresaw, let's say, when I started all my studies back in the very beginning, that I would end up specifically on that area where I combine chemical entity, a small molecule, with a biologic molecule, so where you utilize both of these knowledge as well as expertise. However, looking at things retrospectively, I think I do appreciate that I ended up where I am, where I can really use a lot of my former education, which is, I would say, not a given. Sometimes we have very unlinear paths, which is good to collect experience as well. For me, it really turned out that it was building block, adding different expertise on top of my existing ones, and therefore growing with the modalities.
Amazing. And having worked on these modalities across, different environments, how has your perspective on cancer drug development evolved over time?
For me, it feels always a bit like circles. I mean, we're used to that during fashion, if I may draw the analysis here. Analogy, we always have very favorable modalities, like with ADCs, right? As mentioned, I mean, ADCs were hot more than 10 years ago. It was Kadcyla, one of the first wash-driven ADCs. It was extremely successful. Now is Enhertu, Trodelvy, and the next generation. We do see also clinically blockbusters that are developed here again, and with the recent deal around Araris, Tubulis, so even European-centered companies. In a billion range, we also see very good commercial success of early biotech. So we do see that ADCs in particular are coming back and are circling, and the same will be true for other modalities like gene therapies, for instance, which was on vogue in the 80s and 90s, or in fashion after or during COVID and shortly post-COVID, which is now falls a bit off the flavor of investors. I think it's always a bit circular. Everything is repeating. We do have, of course, some very new modality like immune checkpoint inhibitors. That was an amazing breakthrough that are coming along from, say, every couple of decades. But otherwise, on the protein modality space, we are making fantastic innovation and fantastic improvements, but the general modalities are staying the same.
In the past and where you are now at CIS BioPharma, what do you think makes an ADC succeed?
So an ADC is a fairly complex molecule. I think people are not appreciating how complex that it really is. They always assume, okay, yeah, well, I have my antibody, I have my linker, I have my payload, what could go wrong, right? But in theory, what you're developing are three different parts separately. Then you combine that to a modality. So we have been people very successfully normally by combining either approved antibody with a novel linker payload combination or even novel payload by itself, right? So basically combining and improving one established entity with improvement on the chemistry part, or vice versa, having very breakthrough linker payload that can then be used to further enhance the activity of biologics. Rarely people have developed both at the same time. So having new targets as well as new linker payload technologies that have been clinically validated, I think that is just a bit of too much risk staggering. There have been companies working on that, but the most successful ones always work on one part and then combining that with a more established part.
Okay. And when you are building a next generation ADC, like your current program in lung cancer race, where are you really focusing on innovation?
So we differentiate on the target that we are addressing. A lot of ADCs companies at the moment, and also a lot of very successful ones, are focusing on linker payload development, predominantly also bioconjugation technologies. Now we are working a bit more on the linker payloads, new effective toxins is the next wave that's currently coming and in vogue. And we are working on the wave after, so to speak. So once we have developed our fantastic bioconjugation technology, a stable linker, and effective payload, we need to venture into new targets to address also cancer indication where there are currently no cure or little cure, or if you're running in resistance due to target downregulation, we need to have new modalities that can also be given in a third, fourth, or fifth line treatment for some indications.
So alongside that, you are also working on radiopharmaceutical approaches, particularly using nanobodies in brain tumours. At a high level, how should we think about radiogland therapies compared to ADCs, in your opinion?
So ADCs are targeted chemotherapy, therefore we are calling them chemotherapy 2.0. The same is true for radioligand therapy or radiopharmaceuticals. Radioligands are small molecules that are targeting radioisotope. Radiopharmaceuticals are biologics like VHH. And these are the second generation of radiation therapy, so radiation 2.0. So very targeted, less side effects, and more effective therapeutics.
Okay, perfect. So in terms of what you're seeing in the market now then, what do you think's being backed more with, the venture firms? What do you think people really are wanting to put more of their time into and the money into?
I think what people are fascinated and interesting in ADCs and RLTs are that both classes, while they are complex, are still in the range and realm where we do know potential challenges ahead, like manufacturing and toxicology challenges. We do have a good profile, so the unknown unknowns are minimal. Let's put it like this. You can control the risk. You can estimate the risk, so people have a feeling that there is a good risk estimation. In other areas, which is, for instance, gene therapy or sometimes also TCEs, to a less extent, the risk is a bit less controllable. And in the current investment climate, where we have a lot of geopolitical interesting times, investors are really looking into predictable assets where you can reach a value inflection point fairly soon, where your CMC part, which is very costly, is covered with little uncertainties and paved the way. That's what makes people interested in the first place, that you can estimate risk. And a second, that has both ADCs and radiopharmaceuticals have shown tremendous success in terms of efficacy. I would say, from my perspective, Antibody-drug conjugates are still easier to produce compared to radiopharmaceutical. There we do have a lot of more production challenges still ahead of us.
Yeah.
So that is also a bit the tendency of some investors to cluster around ADCs and to invest in radiopharmaceutical more in the supply chain than in new modalities.
Yeah, I have to agree with you. I think when there's a lot of traction around something like ADCs at the moment, a lot of people tend to follow down that route and rush into things. And what I've heard from speaking to a lot of people, a lot of companies that have failed, is that it's sort of just slinging stuff at the wall as a phrase and hoping that it will stick. And that's why companies do tend to fail. I don't know if you agree with that.
I think there are always, let's say, a multitude of issues why
Yeah,
people
of
can fail. So that's something I learned. It's not always driven by science.
Not always, no. Obviously, you're in the early stages yourself, and your current work is quite an early stage in terms of platform and development. What are the key challenges that you're facing at the moment when building radiopharma approaches?
As pointed out, for us also key challenges are supply chains. I think everybody currently is very eager to get their hands on a limited supply of alpha emitters, especially actinium. That makes it very expensive also to do already preclinical work with these materials because your supply chain is very unreliable. So it's not only more cost intensive than other therapeutic, but it's also that it's not secure that you always get the material on time, and especially when you're planning animal models and you need to inoculate the animals and have a defined treatment window. That can cause quite a lot of stress. If you're not sure that you will get the material in time, so that is a really major challenge to have very reliable partners that can provide the material in time that you can perform your experiments as planned.
Okay, perfect. And then obviously the core debate, which is ADCs versus radioglands. What would you say is maybe one or two strengths of each modality at the moment?
Yeah, for radiotherapeutics, let's start with these ones. And we have mentioned we have both, right? So we carefully picked what are the really strengths of these modalities. And for radiotherapeutics, they're extremely good if you have tumors that are localized, that need to have a high toxicity, fairly concentrated in one area. Therefore, they are amazing, and especially in brain tumors, as we are using them, brain has a high resistance to radiation. That is a very good area. So it's always combining the effectiveness in treating your tumor, so it could be in this case very local, together with the tox profiles. For radiopharmaceuticals, they tend to have a high heme toxicity, so high blood toxicity. Treating brain tumors, for instance, that is not for us a concern. They have a high kidney toxicity. Again, brain tumors are quite far away from the kidney, so that is a very good match for us in terms of efficacy and tox profile. Other way around, are very good for systemic administrations. Administration also potentially for tumors which are more diffused or have more metastasis character, right, where they're distributing throughout the whole brain, because overall comparable with the new technologies, their side effects are very mild. We have seen lung toxicity, however, I can also control that by linker payload and especially bioconjugation development recently. The side effects are very controlled, and you can use them for systemic treatments very effective.
Okay, interesting. And would you say that tumor penetration, is a key differentiator between the two?
Sure. That also depends on the modality that you're using. So with radiopharmaceuticals, you tend to like small modalities because the half-life should be benchmarked below 1 day, so 24 hours, and that you're not accumulating too much toxicity. And for that purpose, they also tend to have a higher, better tumor penetration. So that is definitely a plus with for ADCs, gold standard still is an IgG1 format at the moment, so full IgG, which have at least 10 times larger sizes compared to, for instance, VHH, or even much higher compared to small molecule radiopharmaceuticals. So they're around for tremendous long of time in circulation, which is good if you have metastasis, right? But they are very hard to penetrate tumors. So only 1% will end up in the tumor, and then in the tumor interior that will be even diminished because that is less vascularized systems. Based on the modalities that we have seen so far in both ADCs and radiopharmaceutical, tumor penetration indeed is very different.
Interesting. And then finishing off on our argument, as a CTO, obviously a lot of my network is smaller companies and people starting off in the space. I was just wondering, from your opinion, how do you decide as a CTO where it's best to invest your time and resources, especially when you're working across multiple indications and modalities?
That is a fantastic question, and that's the question where I get paid for. It all boils down to navigating an area with incomplete and incomplete information.
Mm-hmm.
Basically, if we would know which modality or target would be the next blockbuster, I think then biotech will be looking much different than it is today.
Yeah.
So even with the improved tools that we have at our hand with AI that can help us to process the competitive landscapes very effectively and very quickly, and also to connect dots on biology and chemistry side, we still have so little information that it really comes down to a bit of gut feeling, what your risk appetite is, and then also to strategically balance the portfolio of your company. So what we are trying is not to be a single asset company, but to have assets with different risk profiles. The majority have novel target, for instance, but we also have programs where we do have established targets with more twist on the technology side. So we're trying to balance the risk by having various targets and various programs. Of course, that inherently has the risk that you're diluting your efforts on one, right, that you cannot process as effectively. Nevertheless, I strongly believe that picking 2 to 3 programs with very different risk profile and then moving these as fast as possible is in overall much less risky than focusing all of your effort on one single molecule.
So a bit of a looking ahead question then. Fast forward 10 years, where do you see, or how do you see, targeted therapies like ADCs and radioglands changing the way cancer is treated?
That's very interesting. Me as a scientist, what I would of course like to see is that ADCs are replacing chemotherapies. Having said that, for instance, you can imagine combination of Keytruda or ICI immune checkpoint inhibitors plus an ADC instead of plus chemotherapies. And that's also what we do see a lot of large pharma companies doing, a lot of clinical trials in that area. Now, unfortunately, the cost burden of producing an ADC is much higher than producing chemotherapy. So you need to have much improved efficacy profile, much prolonged survival, much more reduced side effects to justify the use of an ADC compared to standard chemotherapy. I think it will really boil down to how cheap can we produce these modalities. If we can produce them cheaply enough that they are competitive to chemotherapy, then there will be the new benchmark. They will completely replace chemotherapy. If we have a 10x difference in production and cost of these modalities, then chemotherapy will be there in 10 years as well.
That's a great insight. Look, it's been amazing to speak with you today and really interesting to explore how two very different scientific approaches are moving towards the same goal. I don't know if there's anything else that you'd like to add, anything about CIS BioPharma?
Yeah, I mean, we are actively looking for new talents. And so we are expanding if people want to approach us to engage. And we're also looking for strong partners to bring our programs forward.
Amazing.
So I think the biotech community is a fantastic community. It's small enough that people know each other. It's large enough that you can always find new connections and everybody's willing to help. So happy to reach out.
Amazing. And are you heading to any conferences this year that anyone can find you at, or any of the team? Yeah,
we are heading to most likely ADC World, of course. Then we are planning JPM to be there. It's next year actually, but there you can find us for sure. And then we will be also around on a couple of smaller companies at conference that we will post most likely via our website.
Well, thank you everyone for tuning in on this podcast. Look forward to catching you on the next.